then next experiment was he put a plate in there and actually still stayed lit. So.
Yep. And plants release oxygen. You're right.
That's cool.
That's right.
That
All right.
Cool.
So, we lost somebody. But Mr. Whitfield, I can't see the screen anymore. So make sure if someone comes in or chat or whatever, you can monitor the chat. And then we'll popcorn read this, everybody. Okay? So this is a little review on photosynthesis, and then a little bit specific on what we're doing in this lab for photosynthesis. So I'm going to start with Whitfield, finish the second paragraph, and then maybe we can read this. Okay. All right, mute yourself for now, everybody. So mute yourself if you're not muted. So there's no other, so we can just hear me, no other background noise. And this is called photosynthesis in leaf discs. And you know what that means in a little bit when I show you. All right, the sun plays an important role to life on Earth. Plants contain chloroplasts that capture the sun, the light energy, and convert it to chemical energy, which is basically stored in sugar and other organic molecules. The name, obviously we all know, is photosynthesis, okay? So plants take the light, convert it to sugar, and then they use the sugar for them to grow and make petals and all that stuff, right? That's how plants work. All right, Mr. Whitfield, next paragraph.
To maintain order, grow, and reproduce, living systems require free energy. Sufficient energy levels are required for individual organisms to grow and also to ensure the survival of populations and ecosystems. Different organisms use various strategies to capture, use, and store free energy. Autotrophic organisms capture free energy from the environment through photosynthesis and chemosynthesis. Heterotrophic organisms harvest free energy in carbon compounds produced by other organisms.
All right, so there's a couple big words you might not know. Does anyone know what an autotroph is? Auto meaning automatic.
Are we autotrophs? Humans?
Does that mean like something produces for you, then you eat it, or?
Yeah. Yes, you're right, Court. An autotroph is a plant that takes the sun's energy and makes its own energy, or something that takes energy itself from pretty much the sun. There's other things like in the deep ocean, you can actually get energy from these smoker vents that come up. But basically, it's using the sun's energy to grab their energy. Okay. Heterotrophs, someone else that's the opposite. A heterotroph then is what?
Everything else.
Everything else. Humans, cows, monkeys, rabbits, chickens, anything that eats something else. So we eat meat and we eat plants. So we don't get the energy from the sun. We get the energy from other things. We have to eat things like plants. We have to eat things like meat. We have to eat things like berries. We have to eat things, on and on and on. Okay. All right. Someone read this. Who can see that well? The next little paragraph.
I'll read it.
All right. Go for it, Hayden.
Photosynthesis occurs in the chloroplast within cells. The photosynthesis process occurs in a series of enzyme-mediated steps that capture light energy to form energy-rich carbohydrates. The overall process is summarized by equation 1 below.
Yep. So this looks very fancy. And basically, if you get into like college biology, you would learn like some intricate steps of this. But basically, all plants do is they take in carbon dioxide. Obviously, they need water, right? Okay. And they need light. So carbon dioxide, water, and light. And what comes out of them? They give us this molecule here, which is glucose, which you guys might have recognized glucose from our diffusion lab, which is a similar thing to dextrose. It's a sugar. And then they actually give us oxygen. Okay. Someone, Hayden, are you still not, maybe me? someone's still logging on. All right. Someone, this longer paragraph, want to read this one? Popcorn, somebody?
I'll pop. Oh, my microphone was on the whole time. I'll popcorn to Mason Corwin, I guess.
Mason, can you see this, Mason?
Yep.
Okay, go for it.
So the net rate of photosynthesis can be determined in two ways: by measuring the production of oxygen, O2, or the consumption of carbon dioxide, CO2. Traditionally, the rate of photosynthesis is calculated by measuring the consumption of carbon dioxide. However, this is difficult to perform with the equipment present in a traditional educational setting. Accurately measuring the production of oxygen is difficult because aerobic respiration occurs at the same time as photosynthesis, thus consuming oxygen as it is produced. Therefore, measuring oxygen production is equivalent to measuring net photosynthesis. A measurement of respiration in the same system would allow the estimation of gross production.
All right. Fancy paragraph, lots of words that just like they jumble together, okay? What we're going to be doing is measuring oxygen, okay, is equivalent to measuring net photosynthesis. So we're going to be doing this thing in syringes, and we're actually going to be like hole punching out some leaves from some spinach and some other things to actually measure the rate of photosynthesis is what we're going to be doing, okay? So this right here is, you're going to be referencing this later on. So I'll read this to you guys. And I have had highlighted in orange because you're going to use this later on in the lab to kind of answer some of your questions. The ratio of photosynthesis, so this equation here, to cell respiration, okay, which basically means when a plant has to grow, it gets energy from the sun, but then it has to use the energy to like make it grow or make the petals open up, okay, or whatever it is. And so it does both. It grabs energy, but also uses energy, okay? And this ratio can be determined by using this. What we're going to be doing is we're actually going to be hole punching little things out of leaves, and they're called them discs, and we're going to have them float, okay? You'll see what we mean when I get to the example. The floating disc uses the overall rate at which oxygen is produced as a measure of a balance between the reactions. So if a plant's got a lot of light coming in, but not really using much of it, then it's like sucking in a bunch of CO2 and giving out oxygen. But if it's growing really fast, you know, and like the petals are moving, then it's using the energy that it got from the sun. So it's like a balance, like how much light and how much energy is it using, okay? So what we're going to be doing is these discs we're going to be making of leaf tissues. This is a fancy word, vacuum infiltrated. We're just basically going to be like pulling a syringe up and down. We're going to replace the air inside the cells with liquid. Okay. So we're going to like make it so like all the liquid, like let's go into the cells is what we're going to be doing. Okay. As photosynthesis takes place, if the rate of photosynthesis exceeds that of cellular respiration, so if more energy is produced than used, okay, it actually will make the leaf thing float, the disc float, okay? But if the rate of respiration or using the energy exceeds the rate of photosynthesis, then oxygen is used up and it's gonna sink, okay? So basically what's happening, if there's more photosynthesis happening, it's gonna float. If there's less photosynthesis happening, it's gonna sink, okay? So that's kind of what's happening, okay? So more photosynthesis means it's gonna what? Float. Less means it's gonna what?
Go down.
Sink, okay? So that's the background information. That background information, I wanted to make sure when you reference it, that's what you need later on, okay? Is that paragraph right there is like the main paragraph when you do the discussion, okay? So now I'm gonna show you a video that what we're gonna be doing, and then I'm gonna show you some of the materials we have here, Okay, so first of all, any questions from what I just shared with you guys? Probably not, because you're probably just waiting for me to go to the next thing, but any questions, let me know. Nada, zip, zero. So, okay, here we go. All right, so here's the video.
In this video, I'm going to show you how to set up your photosynthesis and leaf discs investigation. To start off, make sure you label all your materials. Start by labeling your controls, and then label your experimentals with carbon dioxide. Now you'll notice that mine are also labeled on the bottom, and that's purely for this video. You can just label yours on the side. Once you have your materials labeled, it's time to prepare the solutions. Now each solution is going to get a drop of soap. So the smallest drop possible is what's recommended. So we'll do that to both sides. And then we're going to add sodium bicarbonate or baking soda to our experimental side. And we're going to be adding 0.4 grams to 200 milliliters to make a 0.2% solution. Move some things out of the way. Our next step is to punch out 20 spinach disc leaves, or whatever leaf you're using. Now the key thing is to avoid any of these major veins that you see running through here, and you want to aim for really like the flat spots. There's two ways you can do this. One way is to use a hole puncher. Probably my favorite way is to use a straw. Seems to go a little bit quicker. And so all I'm going to do is I'm just going to push down and twist and start making leaf discs. I need 10 for each syringe. Find another good spot here. Now load these into the experimental with CO2. And then to get them out of the straw, I'll just go ahead and blow. There they go. So they're in there. Obviously with the hole puncher, you can just put them into a little weigh boat and then kind of tip them in there. I'm wearing gloves so my finger oils don't interfere with the discs. You can use forceps also to help you maneuver the discs if you have any issues with the discs. Okay, so the next step, and this is kind of the most frustrating part of this lab, so where students kind of have the most trouble, is we need to get these discs to sink. So our first step is we want to push out almost all the air. We don't want to crush the discs because then it'll mess up our experiment. And this is our experimental with CO2. So we're going to draw up some of the experimental solution, about 3 milliliters, maybe even 4, but definitely not more than half. You'll notice that these are floating right now.
He added too much. He's gonna get rid of it.
That's what we need to change. And so we're going to put this top on, this little stopper. And then
you're only supposed to put like 3 or 4 in there. You put like 7 or 8 in there.
All right. We'll put the stopper back on. And then what we're going to do is we're going to use vacuum infiltration to force the air out of the leaves and replace it with water, which will reduce the buoyancy, making the leaves sink. And the way we're going to do that is we're going to pull down on the stopper. If anybody gets stuck to the side, just pull them down, splash them down. And we'll hold that.
Hold it for about 10 seconds.
Let it go back up. We'll push back up here. And you'll notice that some of them are starting to sink. And you might have to do this several times. If you're having a lot of trouble with it, go ahead and add a little bit of extra soap if it's being difficult. And then, so you'll continue to do that until all of them are sinking. This takes a little bit of time. Again, be patient with it. And again, if you're having trouble, add a little bit of soap. So instead of you guys watching me do that, I have 2 that are done. So you'll do this for both sides. Obviously, you'll suck up the control solution for the control syringe. And so you notice they're good and sunk. Okay, get these out of the way. And to conduct our experiment, we're going to fill these cups about halfway full with solutions.
And not about halfway, you'll need about maybe an inch or
And then
two.
we'll carefully empty the leaf discs into their respective solutions. Much easier. They all should be sinking.
There it goes.
All right, and from here it's time to set up your light. So when you set up your light, the shadow's going to come in. You want to have it about 8 to 10 inches above your leaf discs, above the top of your containers. And to take data, what you're going to do is you are going to turn your light on, and you're going to start collecting data. Now, every minute you're going to count how many leaf discs are floating for a total of 15 minutes or until every leaf disc is floating. And that's all you need to do.
All right, so that's the gist of it. Who has a question just to start out on, like, what are we doing? What is going? Because I'm going to explain some more. But Court, question?
So were they sinking because salt entered the little leaf pod or whatever?
Good question. Not quite. Basically, what's happening is when you, and I'll take my, just want to make my screen.
All right.
When you, can I make me bigger? I can't remember. I don't know. You can see me big? You can.
You have to, it's depending on each person's view.
When you pull this, the syringe, when you pull it like this and it goes up,
Yeah.
it is creating a vacuum, which what's happening is when it happens with the vacuum, all of some of the liquid that the discs are in, it's basically pushing into the cells. And so the more liquid into that little disc,
It'll be heavier.
It makes it more dense, and then it sinks. That's what it's doing. Okay.
So
any air pockets that are inside the cell, it's kind of filling them with the liquid and making them sink. Okay. It's kind of pushing the oxygen out and then filling them in.
Okay.
That's a good question. Okay. Lindy, you had a question? Is it frozen? I can't think what it was. Don't know. Okay. Somebody else question?
Jasmine wants to know when it's due.
It'll be due on the I think the 22nd, which is the last day of grading, but it could be due on the next week. Depends on what the high school team decides for like when grades are finally due. But if we can't get all of it done, I could actually split the lab up into like, you do the experiment and turn that in, and then maybe your first grade for second semester could be like the results and conclusion we might do. We might, if we don't have enough time because of, you know, how things happened last lab, we might split this lab into kind of 2 grades. But it'll definitely, whatever I decide will be due on the 22nd of this month, okay? And I'll explain that a little in what else we have to do. Lindy, you have a question?
Is this going to be recorded? Because
Yep, I'm recording it right now. Yep.
kind of just left the Zoom. So is it going to be recorded?
Yeah.
This is recording right now. Yep.
Okay. Thank you.
Cool. Anybody else before I get to the next thing?
Elijah is asking about leaves.
Okay.
Which leaves we're going to use?
We will get to that next. So good. But a typical one to start out is with spinach. That's the easiest one. Spinach is the best one. You can just get a bag of spinach at the grocery store and do that. Very simple to do that. Okay, but I'll get to those in a little bit and what exactly you guys will be doing.
Are we only doing one type of leaf?
That we discuss next.
Okay.
Because that could be your variable. If you guys are writing up a lab report on this, so you're using all of this procedure, except you're going to have a choice in what you guys can change. You guys can change the type of light to use. You guys could change. You could test out different leaves if you wanted. You guys could test out water temperature. Would cold water versus warm water make a difference? Or you guys could test out the pH of water. You could actually add some vinegar to it or add some hydrochloric acid to it, and that's your choice. Okay, we'll get to that. So you guys will be testing. Okay, good question. All right, any more questions before I move into just what materials you'll be getting? What you have to do?
Wait, what does this do again?
Whatever part that we can get to by January 22nd, which might just be the data collecting and not the resulting conclusion, we'll be doing the 22nd.
So maybe a few days after the 22nd?
Maybe.
Likely we'll have something due on the 22nd or maybe like the 25th. Yeah. So,
okay.
Yeah, my guess, just because I wanted an extra week, would just be that you have to have all your data collected by then, and then your resulting conclusion will be a new grade into the next semester. We'll split the
Okay, I just wondered because I don't really want to be late this time.
Yeah.
Good plan.
That'd be good. All right, so if you have any more questions, let me know after I show you this, what you guys are getting from me. So let me switch my video. Give me a second. View. So I gotta switch my camera, so hold on. Uh-oh, is it not working? It's not recognizing it. Well, that's odd. Sorry, guys. This is supposed to have my other camera so you guys can see what's going on. Give me 1 sec. Let me see if it recognizes it. Recognizing it. Well, I'm going, there we go. There we go.
Nice. There it is.
All right. That just took a lot longer than normal. Okay. So what you'll be getting from us, okay, is if you want tweezers, you could do tweezers to pick up the discs, or you could just get gloves from us. We'll have tweezers or gloves available because these discs that you're going to be creating are super small, okay? But you don't want to touch them with your hands because of oil once you make them. So you can either get some gloves from us or we'll have tweezers for you as well. So these are the one thing you'll be getting. You won't get one of these because you can just use like a bullet home. You'll be getting 4 of these syringes, okay? And they have to be labeled different ways, but this one's CO2. You'll be getting 4 of these and you'll label these, okay? So 1, 2, 3, and 4. You'll get 4 of those, okay? You're gonna get 4 of these caps that go with it, that go on the top of it. If you saw in the video, these caps go right on here. And so it seals it so it doesn't come out. And so then you can suck it out, okay? So you get 4 of those caps. Actually, you're gonna get 5, actually, syringes and 5 caps just in case if it breaks or something. I'm gonna give you an extra one just in case. You're gonna get cups so you can draw on them and label them, okay? So you get 4 cups. And if you need a hole punch, we'll give you a hole punch. We've only got 8 of them. But you could also use a straw, as you saw in the video, to poke them out. So you'll be getting this as well if you need it. I've only got, 7 left because I think Lindy has one. So yeah. Okay. And this is fancy baking soda, but everyone should have baking soda at home, right? Everyone have baking soda at home? Yes, good. So I don't think I have to give you baking soda. I really don't think I have to give you the fancy soap that they gave us. Lindy actually got one, but just in case, because I had to send her the stuff in Michigan before she left. So her and Levi. But there's soap. You'll have to get a ruler, marker at home, and obviously you'll have to get your spinach leaf and poke some holes in it. This is it's actually like a butter leaf that I start poking holes into. So that's a possibility. You can choose the leaves. Okay, I don't think I'm missing anything. The one thing I'm missing, in case you get this hole punch, you know how the hole punches have this part that like collects them? I actually took that part off. Like, so this part right here, okay? I can't even get it back on now. But I took this part off just because when you hole punch them out, they're gonna get collected in here, and so they like actually fall out of here. Okay. All right, so those are the materials that you guys will have, all right? Who has any questions from that aspect? Anybody? Nobody? Lindy is with her entire family.
Yeah, oh my goodness, that is like...
Those are her cousins. And okay, any other questions before I show you the document you guys are gonna need? Nada. Eat a light and see if it sinks in your stomach. What? Dennis, what?
It's like when you eat a watermelon seed and a watermelon plant grows in your stomach.
Oh my goodness. Sorry, I didn't see the chat, so I don't know what you guys are talking about.
That's not true. My mom tried to teach me that for months on end.
It's
Like,
very
"Sophie."
true.
All right, so let me share with you guys the 2 documents that you guys have in your folder now and what you guys need to do with them, okay? So in your folder, which will look like this, you've got that background information, which I shared with you guys, okay? And then you've got this example, okay? So I already have it pulled up here. And so you all should have this shared with you before we leave. I want to make sure that you guys have this, okay? So this is kind of an example of what you guys would be doing and what it's going to look like. You guys are going to be creating a lab report just like the pizza lab, okay? So if you don't remember, your pizza lab is, and I'll get the picture of it, so give me a second to move this thing, has your lab report template. So if I click on pizza and I go lab report documents, everyone's going to have to create another blank lab report so they can start filling this in, okay? So you have to create another one of these for this lab to start out, okay? So that's where you're gonna put this information on there. So what are we actually gonna be doing? And here is the problem that everyone is going to try to solve. How do biotic or abiotic, biotic means basically something that's alive, or abiotic, not living factors in the environment, such as light, pH, or temperature, affect the rate of photosynthesis, okay? So something that is not having to do with life, like light or pH or temperature, how does that affect photosynthesis? Or how does biotic affect? So you could use those different leaves like you asked Hayden. How does something that's living, how do different plants do photosynthesis? Maybe spinach does it faster or slower than butterleaf, then different than kale, then different than, something else, okay? So you could test out something that's alive or not alive as your variable. Our variable in pizza was more flour, more sugar, less sugar, more rise time. You guys are gonna adjust this lab to test something that you guys want to test. Okay?
So, real quick for everyone, a lot of you guys kind of messed this up in the pizza lab. Everybody has the same problem. So the problem that's written right there is everybody's problem. Your hypothesis is unique to you, but the problem is the same for all of you.
Exactly. So let me kind of walk you through really quickly, because we've done the pizza lab, so I'm not going to go in detail, but really quickly what you should be doing here. So let's say if I use spinach and butter lettuce, that's the ones I have here, to test the rate of photosynthesis, then let's say you think spinach will have a faster rate, okay? Because maybe a reason is because it's darker and contains more chlorophyll, okay? Because chlorophyll is the pigment that soaks up the sunlight. So maybe you think because it's darker, it contains more chlorophyll, that's what's gonna, you know, have it be more photosynthetic. So that might be your hypothesis, okay? So this is what you're grabbing from me, okay? Oops, I should have said tweezers or gloves, I forgot that. Not if needed, we wanted to grab those. Okay, so for me, cups, syringes, syringe cap, and tweezers or gloves. Everything else you should be able to do on your own, okay? Dish soap, baking soda, which is called sodium bicarbonate. Mr. Whitfield actually moved it to a half teaspoon. I did some more research, but it's a half teaspoon of baking soda you're gonna have to be measuring. Water, hole puncher, straw, paper towels and napkins for cleanup, permanent marker and tape, a ruler, timer, and a drinking glass. Pretty simple, okay? So here are some things that you could change, okay? So the type of light. So if you wanted to use just spinach, but test out a grow light versus a fluorescent light versus just the normal light bulb, you could do that and test out the different rates. If you wanted to test out water temperature, you could do that. Hot or cold water, does that affect it? Do you want to make it more acidic? Add vinegar or hydrochloric acid. Or do you want to test out different leaves? I didn't do that one yet.
If you're testing temperature, it'd be best to have a really, like a thermometer and get accurate.
Yep. You get a thermometer then. Good. All right. So who has just a question about hypothesis and like kind of just the general aspect of what you guys are doing? Okay, moving on. All right. So this is the procedure that you guys will be doing. Now, just like the pizza lab, you're gonna be doing a control, which the control is already in the directions. You're gonna have to adjust something here that you're gonna be testing. So most of this you're gonna be copying as your procedure, but you're gonna be adjusting what you need, okay? So this is preparing the cups, solution, and syringes. It was explained in the video, and you can follow it step by step here. It's not too hard, okay? Adding leaves to the syringes, and so you have to punch them out, and then as you saw, you have to put them in there, okay? Adding solutions. I'll make that another, okay? So we're adding the solution to syringes. You know, you basically suck it up. That's what they did there, okay? And then this could be something that's different. So when you transfer it, Anything in yellow is something that you could have changed. So like the sodium bicarbonate, do you want to add more like vinegar to that instead to make it so it's a different solution? But why would you want to add vinegar? Why would you want to add pH to it? Maybe it's because you live in Bend area and there's not as much soil, it's more sand, it's more acidic, okay? Do you want to change the light source? That's the thing that you want to test, okay? I forgot to actually change the types of leaves. You can actually change the leaves if you want to test that too. So that was one thing I forgot to do in the yellow. Okay, so you have to figure out what you want to test, okay? And that's highlighted in the yellow, okay? Once you are done with this, you're gonna create a data table very similar to this. Let me make it a little bit smaller. So your data table is gonna look like this. Remember how we said that once you're done with it and you put it in the solution, you're gonna see how many float every minute, okay? And in this example, we used spinach with CO2, okay? Sorry, whoops, I just switched that right. Yes, leaf type 1, so spinach would be what I would be doing. And then we'd have control spinach, okay? And then we would have butter leaf, butter lettuce, sorry. And then I would also have butter lettuce. Okay. So that would, if I was testing different types of leaves. If you want to test types of leaves, I want you to have you actually do a third one, okay? You could possibly do a third one if you need to, okay? And then we'll get to this later on. I don't think we're going to be able to get to the questions and everything later on for your results in conclusion. I think we'll probably just do the data table and the graph before the break, for like the end of the semester break, okay? So I think I'm just gonna have you guys do the data table. We're gonna create a graph, which we can help you in a couple weeks. And I think that'll be it for this grade, because we've only got 3 weeks to do it, and you guys are picking up the materials next week. So it limits us to only 2 weeks to do it, okay? So that is what you guys be doing. Who has a question? Clarifying, anything?
Jasmine.
Do we have to use both butter lettuce and spinach, or can we just use like spinach or just butter leaves?
That's up to you. What do you want to test? Do you want to test out different leaves that do different photosynthesis? So then obviously you'd have to choose 3 different types of leaves to test them out. Okay. Do you want to test the light? So then if you want to just test the light and how that affects photosynthesis, then you just only use spinach or only use butter lettuce or only use kale, whatever you want to use. Okay. But whatever you choose, you obviously have to, everything else has got to be the same.
Okay.
Yeah. So again, you can test different lights. So I've got a light right here. Let me show you. This is, some of you have like chickens at home and animals. This is like a hot lamp. So if you wanted to test light, you could test like a normal light bulb and like your lamp. This could unscrew and go to like a normal, lamp. So this is like a heat lamp. If you have a black light at home, that could test it out. Obviously, different lights you could test out for different photosynthesis. Okay. If you want to test out different leaves, then you've got different leaves to test out, and everything else stays the same except for you're doing different leaves. Okay. If you want to test water temperature, then you have to pick different temperatures: ice cold, normal, lukewarm, what you feel like you want to test. And if you want to test like pH, like how acidic it is, you could add some vinegar to it or add some acid to it, which I could give you guys some acid, as long as you guys don't drink it because you'll get killed. No.
Mr. Jackson, could you clarify what we're measuring?
You are measuring how quickly those discs rise. And if you remember back in the information, the background information, when you pull that syringe, you are sucking out the air from the cells, okay? So what you're measuring, as soon as you put the light onto the discs, what's gonna start? Photosynthesis, okay? You're starting to measure how quickly photosynthesis happens, okay? Versus you actually cut this, like the little leaf thing, so that might even be trying to like repair itself, even though it's like a disc. But cells inside there, each cell might think it's kind of still alive, so it might try to repair itself and use the sugar in there to try to repair itself. So you're measuring, is the cell using energy or is it making energy by photosynthesis? That's what you're measuring. You're measuring how much photosynthesis happens, which makes it float. Okay? You're measuring how quickly photosynthesis takes place. Okay? Any more questions? Lots of talking by me, I know.
So
the lamps are not for keeping the leaves alive. The lamps are just for making photosynthesis happen.
Oh.
And
Like,
How long should this experiment take? As in like half an hour or 3 weeks?
I would say the whole thing, setting it up will probably take you about a half an hour. And then depending on how many of these you do, you've got to have it, you know, float up, and you could do all the discs, all the cups at once, but it's going to be really hard for you to be able to, you know, look at all 4 cups every minute. So it's probably going to take you 15 minutes for each cup. You know, you have to wait for it to go up. So I would say overall a couple hours, you would say, you know, from the setup to like watching the cups float up. So
I'm quite confused over this because what do you mean by photosynthesis? Like it gets bigger or?
Nope, doesn't get bigger. It's just producing oxygen when photosynthesis.
So it's just
oxygen.
so when the leaf discs go in water, if it's producing oxygen, it's going to float because oxygen floats. If it's not producing oxygen, therefore not doing photosynthesis, it'll sink.
So do we necessarily have to have a heat lamp or?
Nope, you just have to have some type of light source that can get pretty cold.
Each experiment will have, you know, the standardized control things, but if you choose to change that variable for your variable of light, then you'll have different lamps.
Yep.
Could you use purple light?
Sure.
You could, but if you're using purple light, definitely that would be your variables on you using different types of light. Yeah, yeah. So here's what you guys need to do. You guys, because I need to tell this to all the labs, you can start picking up materials from us next week, and then we want you guys to, and we'll remind you guys next week, but we want you guys to pick up the materials next week and start doing the lab next week. But before you pick up the materials, what you need to do is make a hypothesis, okay, and then adjust your procedure. Okay, what are you doing? Adding more leaves, adding different lamps. You know, you have to modify my directions to make it fit to what you guys want to test. Make sense? So you have 2 things to do, actually 3. Make a new lab report. That's 1. 2, make your hypothesis. And 3, adjust your procedure and your materials to what you need. Okay? All right. So I wanted to end 5 minutes early because I know some of you have other classes. I'm so sorry. I didn't check the time. I know somebody had 11 o'clock. So watch this video if you have any questions, and email us if you have any questions as well. But we will be in contact and next week on where to pick the materials on Seesaw. So check your Seesaw. Okay.
Okay.
We'll send it out probably Friday.
All right. Bye.
All right. See ya.
Bye.
Yeah.